Hi, welcome to today's AP Biology lesson. Today our lesson will review material in Unit 1. Just to remind you that on Friday, April 3rd, an announcement was made providing the details about the 2020 AP Biology exam. The 2020 exam will be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. All exams will be taken by students at home. The exam will consist of 2 free response questions. The first is a long FRQ and will have a 25-minute time limit. The second question is a short FRQ and will have a 15-minute time limit. Beginning today, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. I just wanted to share with you a resource you can find at AP Central. The first thing I want to point out is at the top here where it gives you the time. That is Central Standard Time, and I reported to you that the exam would be administered on Monday, May 18th at 2 p.m. Eastern Standard Time. So you need to figure out what time zone you're in to know what time your exam will begin. There will be a makeup on Wednesday, June 3rd, and that will be for students who are unable to attend the primary test date. And as I said, there will be 2 questions. I'll talk a little bit more about the specifics in the next slide. And at the bottom here, you can see the link to our daily course reviews from 4 to 4:45 p.m. Monday through Friday. And if you're already here, you don't need the link, but if you want to share that, you can. So just to remind you about the structure of this year's exam, the first question, which is a long FRQ, will be about interpreting and evaluating experimental results. You will have 25 minutes to write your response and an additional 5 minutes to upload it. This will make up 65% of your total exam score. Now, in previous test years, when I'm preparing my students, I usually tell them that they should plan on spending between 20 and 22 minutes on a long FRQ. So we're right in that area with a little bit more time this year to actually read and then record your response. So what will happen in this first long FRQ is you'll be given a scenario accompanied by data that is presented either in a data table or a graph. And then this is where our task verbs, those of you that have been joining us know that we talk about the task verbs in each of our lessons, and that's where these come into play. So you will be asked to describe and explain biological concepts, processes, or models. You will be asked to identify experimental design procedures, and you will be asked to analyze data as well as make and justify predictions. And you can see in the far right column the number of points that you would be awarded for each of those sections. For the second short free response question, in previous exams when I'm preparing my students, I usually tell them that they should spend between 6 and 8 minutes on those short free response questions. This one is a 4-point question, and you see that you have 5 minutes to respond to it and then the additional 5 minutes to upload your response. This will make up 35% of your total AP exam score. In this question, you'll be given a scenario describing a biological phenomenon with a disruption. And again, here are task verbs. You need to describe biological concepts or processes, and we're going to actually practice that today. You need to explain biological concepts or processes or describe characteristics that are represented visually. And if you were with us last week, a really good example of a concept that is represented visually is phylogeny, and we talked about how to construct phylogenetic trees and cladograms. You also need to be able to predict the causes or effects of a change in a biological system and then justify those predictions. And so each one of those sections is equal weight with 1 point each. I just wanted to share with you a slide that indicates what the University of California system, and I suspect many other colleges are saying about this year's exam. The College Board recently announced the changes to the AP exam content and formats for the spring 2020. UC recognizes the effort that students have already applied in these challenging courses and will award UC credit for the 2020 AP exams completed with scores of 3, 4, or 5, consistent with previous years. So this should be reassuring that even though the exam format has been changed, your scores will still be awarded college credit, at least in the University of California system. And as I said, I expect other colleges to put out statements soon. So I'm Allison Kate. I'm affiliated with both UC Berkeley and Redwood High School, and I'm teaching today from my home in El Cerrito, California. So let's do our warm up. If you were with me yesterday we talked about ecology, and I gave you a homework practice FRQ about invasive species. So if you have that homework response, go ahead and get that out. If you'd like to, if you're just joining us and you'd like to try to answer this as we talk about it, then get that pen and paper handy and please do so. So the question says, invasive species such as red fire ants introduced into an ecosystem often threaten native plants and animals. You are asked to describe 3 different factors that contribute to the success of invasive species in an ecosystem. So let's take a look at how this was scored. And this was worth 3 points, and each bullet point is worth 1 point, and you can earn up to 3 points. So any one of those responses would earn a point. So saying things like, since there are no natural predators, parasites, or pathogens, the invasive species will be successful. They may have an effective aggressive mechanism to be able to be successful. They have no limit on their resources. There are no environmental inhibitors such as pollutants. They may be an R-selected species, which means they have an increased reproductive capacity and they grow really rapidly in terms of their growth rate. They may introduce a new phenotype into a large population that can be selected for if it's advantageous. They may be occupying a niche that is not occupied by any other species, so they have no competitors. Prey may lack defense mechanisms because they haven't encountered this new species, and they may have favorable or appropriate environmental conditions such as rainfall and temperature and sunlight and so forth. So if you selected 3 of those or wrote those 3 factors, then you would have scored a 3 out of 3. Good job. All right, so today, what are we going to be working on? As I mentioned, it's our first review session. I'm sure you're excited to get to the review. We're going to take you way back to the chemistry of life. This is in Unit 1, and we're going to be reviewing 4 topics and 1 science practice. So today, we're going to look at the structure of water and hydrogen bonding. We're going to look at the elements of life. I'm going to introduce you to biological molecules and properties of biological molecules. And today, I'm going to focus on the carbohydrates and lipids. And tomorrow, you'll get a lot of instruction on proteins and nucleic acids from Ms. Pfizer. We're also going to be looking at Science Practice 1A, which is asking you to describe biological concepts or processes. So if you think back to that second FRQ, that is one of the task verbs. Today, we'll be looking at several free response practice questions that combine the content of each of these topics and skill. There are some terms that hopefully when you hear them, you'll remember them from prior lessons. They are dehydration synthesis, hydrolysis, monomer, and polymer. So we're going to talk about those concepts today. Using those terms. By the end of the lesson, here are your objectives. You should be able to explain how the properties of water that result from its polarity and hydrogen bonding affect its biological function. You should be able to describe the composition of macromolecules that are required by living organisms. And you should be able to describe the properties of monomers and the type of bonds that connect monomers in biological molecules. And I might note that each of the type of bonds is different depending on the class of biological molecules, but they are all created and broken down using the same chemical reactions. So here's what you need to know. Just want to kind of bring you back to where we are in the framework. I told you I'm taking you way back to Unit 1, Chemistry of Life, and units are broken down into topics, and each learning objective is what students need to be able to do with the content knowledge for that topic. The essential knowledge is the knowledge that's required for you to perform the learning objective. So we'll be reviewing that on our debrief. So you can see that we have 4 topics in Unit 1, and the rest of those topics will be reviewed tomorrow. So let's go ahead and start with the content. So in topic 1.1, we're talking about the structure of water and hydrogen bonding. And the idea here is that the subcomponents of biological molecules and their sequence determine the properties of that molecule. So I want to start with going way back that everything is made of matter and matter is made of atoms. And so I have a little cartoon for you. Hopefully that'll give you a little giggle. Since water is made of hydrogen and oxygen, we'll kind of review atomic structure using those 2 atoms. So I've got a hydrogen atom, which contains 1 proton in the nucleus, and protons are positively charged, and 1 electron outside of its nucleus in its orbital. We also have oxygen. Oxygen has 8 protons, which are positively charged, and 8 neutrons, which have no charge, and those are located in the nucleus. And then we have 8 electrons. 2 of the electrons, which are negatively charged, are in the first energy level, and the remaining 6, or valence electrons, are in the second energy level. And just to remind you that it is the electrons that determine the type of bonds that are going to be formed. So the number and arrangement of electrons. So we'll take a look at that in the next slide. So when we talk about water, molecular formula is H2O. It's composed of 2 hydrogen atoms and 1 oxygen atom, and they're bonded together with covalent bonds. To remind you, covalent bonds share electrons. I'd also like you to notice that there is a negative sign around the oxygen and positive signs around the 2 hydrogens. And we're going to talk about that in more detail now. So when we see those symbols, and in this image, we see this funny D right here, which is the Greek letter delta. So this delta is the symbol for a partial charge. And you can see that in the water molecule. It has 2 partial negative charges and 2 partial positive charges. And if you notice that the negative charges are around the oxygen, where the positive charges are around the 2 hydrogens. And the reason for this is oxygen is more electronegative, so the electrons are shared unequally. So the oxygen ends up hugging the electrons, and they spend more time around the oxygen, creating a slightly negative side, and less time around the hydrogens, so they have a slightly positive side. We call this being polar. So water is a polar molecule. As a result of water's polarity, there are special bonds called hydrogen bonds that form. And hydrogen bonds don't just form in water, although that's what we're going to be focusing on today, but hydrogen bonds form in many other biologically important molecules. You'll see tomorrow that hydrogen bonds are found in proteins and are responsible for their secondary structure. And you'll also see tomorrow that in DNA, the 2 strands of the DNA helix are held together by hydrogen bonds. But right now, I just want you to see that what happens is that the negative side of one water molecule is attracted to the positive side of the hydrogen in another water molecule. That is a hydrogen bond. And hydrogen bonds are often represented with those dots, so you can tell that they are connected. The other thing, and you'll see in a later image, one water molecule can form up to 4 hydrogen bonds with other water molecules. So as a result of water being polar and forming hydrogen bonds, there are a number of properties of water that emerge that are very important to life on Earth. I'm going to go into more detail about the first 3 that you see there, and they are cohesion, surface tension, and adhesion. But I'd also like to mention high specific heat, high heat of vaporization. Water is a versatile, in fact, termed the universal solvent. It is excellent at dissolving polar molecules. It has an unusual phase change, which I'll explain in a minute, and water ionizes. So let's go back to high specific heat and high heat of vaporization. It means that because of these hydrogen bonds, it takes a lot of energy to change its state of matter. So it takes a lot of energy to get it to go from a liquid to a gas, and that is the high heat of vaporization. It also means that water resists change, the high specific heat. It takes a lot of energy to change water's temperature. And you're probably familiar with that if you live near a large body of water. I live right by the San Francisco Bay, and the water will moderate our coastal temperatures, whereas inland, farther away from the water, you see a much more wide range of temperatures. It gets hotter and colder. So both high specific heat and high heat of vaporization are due to hydrogen bonding and the fact that it takes a lot of energy to change its state of matter or its heat capacity. Unusual phase change is also due to hydrogen bonds. And what I mean here is that when water freezes, it goes from a liquid to a solid, it actually becomes less dense and ice floats on water. And this is not true for other solids. Other solids become more dense. So this creates some really advantageous things for living things. For example, if you're a fish and you live in a lake, that lake will not freeze solid. The other thing, it allows seasonal snowmelt. So the fact that water does become less dense when solid is due to the way that those hydrogen bonds form. And finally, a word about ionization. When water ionizes, it produces hydrogen ions and hydroxyl ions. And you may be familiar with the pH scale. It is a measure of how many hydrogen ions are in a solution. And if there are a lot of hydrogen ions, it has a low pH. It's very acidic. And if it doesn't have as many hydrogen ions, but has more hydroxyl ions, we say that its pH is basic. If the amount of ions are exactly equal, we call that neutral pH. And most of the chemical reactions in living things do occur at a neutral pH. There are some exceptions, and I believe when you get into talking about enzymes, pH will be talked about in more detail. So let's talk about those three properties that result from hydrogen bonds in a little bit more detail: cohesion, adhesion, and surface tension. Cohesion means that water molecules like to stick to each other through hydrogen bonding, whereas adhesion means that water molecules like to stick to other molecules through hydrogen bonding. And so if you look at the pictures I have for you, cohesion is responsible for the water beating up. You see that on the leaf. It's also responsible for beating up on that spider web, but it also adheres to the protein in that spider web. So that image shows you both cohesion and adhesion. And then finally, surface tension results from hydrogen bonds resisting being stretched. So it creates a nice tight surface. And the image there is showing you a water strider that can actually walk on water. And if you've ever skipped rocks on a lake, you know this feature of water. And just again, reminding you that if you look at the diagram here, it's kind of reminding you why these things happen, that there are hydrogen bonds, and this water molecule is forming hydrogen bonds with 4 other water molecules. And that is kind of the maximum number that the water molecules can account for in hydrogen bonding. So moving on to topic 1.2, we have the elements of life. And all organisms must exchange matter with the environment to grow, reproduce, and maintain organization. Atoms and molecules from the environment are necessary to build new molecules. So we're going to focus on carbon, nitrogen, and phosphorus right now. But if you see, I've got tiles from the periodic table, and you may have learned this as CHNOPS: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the 6 elements that make up 96% of living things. The other 4% is made up of some other elements such as chlorophyll and calcium and potassium. And so they are important, but they are way less prevalent than these 6. So carbon, and we're going to talk more about this, but carbon is used to build all of the biological molecules, carbohydrates, proteins, lipids, and nucleic acids. Carbon is used in storage compounds like starch and glycogen, and also in cell formation in all organisms. It forms the cell wall in the form of cellulose in plant cells. Nitrogen is used to build proteins and nucleic acids, and phosphorus is used to build nucleic acids and certain lipids, the phospholipids. More on all of this is coming up. So I want to introduce those terms I told you should be familiar with it just in case you forgot. And these are the terms monomer and polymer. Monomer is the subunit that serves as a building block of a polymer. So mono means one, poly means many. So a polymer is a larger molecule that's made up of many similar or identical monomers that are linked together by covalent bonds. And I'm going to give you examples in each of the groups of organic molecules. So the monomers of carbohydrates are monosaccharides, which are also known as simple sugars. And you're probably familiar with glucose, but there are other simple sugars that I'll point out in just a moment. And I'm showing you the ring structure of glucose. The monomers of proteins are amino acids. And I just have a generalized image of an amino acid there. We'll talk more about what those components are in just a minute. And finally, the monomers of nucleic acids are nucleotides. A nucleotide is composed of three parts: a phosphate group, which is why we need that phosphorus, a pentose sugar, and a nitrogenous base. And that nitrogenous base is why we need the nitrogen. So let's take a look at carbon. This flow chart kind of shows you why carbon compounds are so important to living things. They make up carbohydrates, which consist of sugars and starches. And as I mentioned, cellulose, which is a structural component made of carbon, hydrogen, and oxygen. They are often called hydrates of carbon. Quite literally, there's twice as much hydrogen as carbon and oxygen. We also have lipids. Lipids consist of fats, waxes, oils, and steroids. And what they all have in common is they are nonpolar, and they are composed of carbon, hydrogen, and oxygen. And in the case of phospholipids, phosphorus. Nucleic acids consist of nucleotides, which contain carbon, hydrogen, oxygen, and phosphorus. And proteins consist of amino acids, which contain carbon, hydrogen, oxygen, and nitrogen. So I want to talk a little bit about the two reactions that are really important in building and breaking down our macromolecules. And these two reactions are called hydrolysis and dehydration synthesis. And you probably, from those names, hydro and dehydration, understand that they have something to do with water, and you would be correct. Okay, so dehydration synthesis literally means the dehydration or removal of water. And the way this works, if you look at the monomers, you've got a monomer which has a hydroxyl or OH group indicated in blue, and you've got a second monomer which has a hydrogen that is showing you, it's an OH group, but the hydrogen is in blue. And so what's going to happen is that we're going to remove the water by combining the hydroxyl from one monomer and the hydrogen from another, and that forms water. And then the resulting monomers are linked together to form a polymer, and they are linked together by a covalent bond. Okay, I'm going to give you specific examples with macromolecules in a minute, but this is kind of a generalized view of dehydration synthesis. I should also tell you that dehydration synthesis is known as condensation. And the way I like to think about it is you're condensing those two monomers by removing water to create that polymer. The opposite reaction is called hydrolysis, which literally means using water to lyse or break apart a larger molecule into smaller molecules. So if we've got that polypeptide or polysaccharide or nucleotide that is linked together, if we add water, we hydrolyze it, we're going to break that bond, and we are going to split those monomers into two and re-add that water to each monomer. The hydroxyl group will go to the first one, and the hydrogen will go to the second one. So let's take a look at hydrolysis in actual biological molecules. So I've got a disaccharide, which is a 2-unit sugar. So it's composed of 2 monomers. And my example is sucrose, which is probably what you put in your coffee or tea. And it's cane sugar. So we want to break that down so that we can digest it better. By the way, disaccharides are kind of how the sugars travel around. They're the transport forms of sugar. And we want to break it down. It does require enzymes and energy. The enzyme in this case is called sucrase. We add water, we're going to hydrolyze it. And when we do that, we are going to break it down into 2 glucose monomers. Okay, well, excuse me, in the case of sucrose, we would get 1 glucose and 1 fructose. In the case of maltose, which is another disaccharide, you would get 2 glucose monomers. We can also hydrolyze proteins. So what I'm showing you is a dipeptide, and what we're going to do is we're going to add water to bust apart that carbon-nitrogen bond right here, which is called a peptide bond. And I'm going to show you in a later slide how that bond actually gets formed through dehydration synthesis. But we add the water, and we end up with 1 amino acid and another amino acid. If this was a polypeptide, we're only showing you 2 parts. It actually is, because here's another peptide bond. This is going to be the peptide, and this is the amino acid. But you can see that what happens to the water is the hydroxyl group goes on this end, and the hydrogen goes on this end. And I'm going to give you some more information about what these ends are called in a couple of slides. So let's take a look specifically at nucleic acids and proteins in just a minute, because the structure and function of polymers are derived from the way their monomers are assembled. In nucleic acids, biological information is encoded in the sequence of those monomers. In other words, each nucleotide, which in turn has that 5-carbon sugar, a phosphate, and a nitrogenous base, the sequence of those is going to determine the biological information. And just a note that DNA and RNA differ in both structure and function. And as I mentioned, you're going to get a lot more information about this tomorrow. In proteins, the specific order of amino acids in that polypeptide, which is known as the primary structure, the sequence of those amino acids determines the overall shape of the protein. Amino acids have directionality. One end has an amino group and the other end has a carboxyl group. And I'm going to illustrate that with an image in just a minute. The R groups, which are called the functional groups of the amino acids, can be characterized by their own chemical properties. So they can be classified as hydrophobic, hydrophilic, or ionic. And the interactions of these R groups determines the structure and function of that region of the protein. And again, you'll get much more on this tomorrow. So let's take a look at how we build a nucleic acid. So we are going to do a condensation or a dehydration synthesis reaction here. And so what you've got is 1 nucleotide. It has its phosphate group, and there's our phosphorus. It's got a pentose or 5-carbon sugar, and it's got a nitrogenous base. And we are going to remove a water molecule. We're going to remove a hydrogen from this nucleotide and a hydroxyl from this nucleotide. That will form water, which you see over here, and those 2 nucleotides will be linked together through a covalent bond, and this is called a phosphodiester link. This is just showing you that nucleotide. It's composed of a phosphate, a 5-carbon sugar, and a nitrogenous base. And this just kind of puts it all together for you with the overall structure of a nucleic acid. Since this has 2 strands, it is going to be DNA, and I also know that by the 4 types of nitrogenous bases here. One other thing to point out is you can see those 2 strands of DNA are connected by hydrogen bonds. So hydrogen bonds don't just occur in water. Now let's take a look at how monomers of amino acids are put together to build a protein. This is the generalized structure of an amino acid. You can see that it starts with a central carbon, and remember that carbon has 4 covalent bonds available. And it's got an amino group or terminus, NH2, and it's got a carboxyl group or terminus. Then it's got a hydrogen that takes up its third covalent bond, and the R group is the side group. This is interchangeable. So each of the 20 naturally occurring amino acids has a different R group, and that will determine the characteristics of that amino acid. It can be simple like a hydrogen, which is the case of glycine, or it can be a complicated ring structure, which you might see in an amino acid called phenylalanine. So let's take a look at a condensation or dehydration synthesis reaction that is used to build a polypeptide. So here's our first amino acid, and here's our second amino acid. And again, we've got our amino end and our carboxyl end. So what's going to happen, and this is kind of funny, but what's going to happen is the hydroxyl from the carboxyl group is going to join with the hydrogen from the amino group to form water. When that happens, notice what gets bonded together, the carbon and the nitrogen. And that carbon-nitrogen covalent bond is found only in proteins, and it is called a peptide bond. That's a really important thing to remember. So let's take a look at some carbohydrates and see how their structure and function is derived from the way that they're assembled. So again, just to kind of give you a flow chart, over here we've got our group, which is the carbohydrates. They can be either simple or complex, and you might know this from your own diet. If you eat simple carbs, you're going to get readily available energy, and that comes from your monosaccharides, which are glucose, fructose, and galactose. And disaccharides are kind of the way that they're transported around your body or a plant body. It's how the organism kind of moves their sugars from one place to another. And there's maltose, lactose, and sucrose. And you may be familiar with lactose. It's milk sugar, and you may be familiar with it because you've heard of something called lactose intolerance. And that means that individuals who are lactose intolerant do not have a functional enzyme lactase to break it down using hydrolysis. So more on that another time. I'm sure that will come up again. So if we look at the image where I've got starch versus cellulose, right? Starch versus cellulose is all in how those monomers are assembled. And what I would like you to see in starch, that is a storage form or a complex polysaccharide. Plant starches are a little bit different than animal starches. Glycogen is an animal starch. And in this case, when this flowchart is talking about fibers, we're mainly talking about cellulose, which is the bottom image. So I'm sure you can notice that there's a difference between starch and cellulose. And if you look I'm going to draw an imaginary plane across these ring structures. The bond is below that plane. And it turns out that when we link our glucose monomers together using dehydration synthesis, if that bond occurs below that plane, we're linking alpha glucose monomers, and we can easily digest those. We have the enzyme that can break those bonds and release energy, and so we can use that for an energy providing molecule. If you look down here at cellulose, if you look at that imaginary plane, the bond is above the plane. And the reason, and the way I remember that is these are beta glucose monomers that are linked together. And I remember this as beta above. And it turns out that humans do not have the enzyme that recognizes that what we call a 1,4 beta glucose link. And the reason that we can't digest cellulose is we don't have that enzyme. So cellulose makes up the bulk of our waste products because we can't digest it. Of course, there are organisms that can. All you need to do is think about termites, and they can get their energy from eating wood. So let's take a look at one more example of the carbs and their sugar monomers. In this case, we're showing you another dehydration synthesis or condensation reaction. In this case, we have two monosaccharides, glucose and glucose, and we're going to build the disaccharide maltose. We're going to do that by removing a water molecule. When we form this, this is that 1,4 link, and this is called a glycosidic linkage. Remember that this reaction, dehydration, because we're removing water, requires energy and the correct enzymes to build that disaccharide. And then you would just keep doing that to build your polysaccharide. I want to talk about the last group of biological molecules that I'm going to get into detail about, and those are the lipids. Again, let's start with this flow chart here. So lipids are all grouped together because they are nonpolar, and you've got fats, sterols, and phospholipids. Fats are composed of triglycerides, and I'm going to show you how you can build a triglyceride using dehydration synthesis. Triglycerides are stored fat or consumed fat. They make up cell membranes. Specifically, phospholipids are important in cell membranes, and I'm going to go into detail about that. They're also extremely important in creating the myelin sheaths that cover your nerve axons. Sterols are very important. Sterols are steroid molecules which are characterized by being nonpolar, which is why they're classified with the lipids, and they are 4 linked carbon rings. Probably the most familiar one you know is cholesterol, and cholesterol is important in cell membrane structure. It helps to maintain the fluidity and provide structure for that cell membrane. We tend to think of it in dietary terms, and a lot of people think it is bad. And while it is, in fact, there is one type of cholesterol that is not good for you in terms of your blood vessels. Your blood is polar, and these cholesterol molecules are nonpolar, and they tend to get stuck to the inner lining of your blood vessels, and that can cause problems. But cholesterol is a really important molecule. It is the basis of your sex hormones, testosterone and progesterone and estrogen. It's also the basis for vitamin D. And then finally, phospholipids. Phospholipids are super important as part of every lipid bilayer, which makes up cell membranes. So that's kind of the breakdown of the lipid flow chart. They are non-polar, and that's an important thing to remember as to why they're linked together. There are 2 things I want you to know. Differences in saturation determine the structure and function of lipids. So you've probably heard in terms of diet that there are saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have no double bonds. They tend to be solid at room temperature, and they're more animal fats. In terms of your diet, they're less healthy for you. The unsaturated fatty acids do have double bonds. You can see that that is indicated by the green portion of that molecule, which means that they're a little bit more flexible. You can also have polyunsaturated fatty acids, which have more than one double bond. These tend to be your plant fats and or oils. They tend to be liquid at room temperature. And then finally, phospholipids. Phospholipids are really important molecules because they contain polar regions due to that phosphate. And you can look up here, the phosphate is shown in blue, and it's hydrophilic, meaning that it likes to be near water. Hydro means water, philic means affinity for. But it also, because of the nonpolar fatty acid chains, is hydrophobic at the same time. And this makes it a useful molecule in creating that barrier as part of the cell membrane. So I've showed you this cell membrane. It's not a great image, but you can see that this is the phospholipid bilayer. The phosphate heads are at the top, and they point both into the cytoplasm and out to the surface of the cell membrane. And then we've got all of these fatty acid tails, which are pointing toward each other because they're hydrophobic. One other thing, you can see a cholesterol molecule is embedded here, and that gives it a little bit of fluidity. You can see one here, it's not labeled, but that's also a cholesterol molecule. And then one other key component, as long as we're talking about cell membranes, is proteins. And proteins can span the entire cell membrane, or they can be on the surface. And those are called peripheral membrane proteins versus the integral membrane proteins that you see there. Okay, so let's talk about how you build a triglyceride. So I'm showing you a fatty acid. In this case, it's palmitic acid, and you can see that this is a saturated fatty acid because there's no double bonds. You can also see that it's got this carboxyl group at the end, and it's going to give up a hydroxyl group. And then we've got a glycerol molecule, which is a 3-carbon alcohol, and it's going to give up a hydrogen when we use dehydration synthesis. To build a triglyceride, you need 3 fatty acids. So what's going to happen is we're going to do that, and we're actually going to remove 3 water molecules. So you can see that that is a triglyceride. You've got the 1 glycerol molecule, and what happens is this bond here is called an ester link. So where the water was removed in the dehydration synthesis, you get that bond, which is called an ester link, and that is an example of a triglyceride. Let's get some practice. And I have 4 practice FRQs, 1 for each topic. I'm not sure we're going to get to all of them, but if you want to go back and look at these, once this is recorded, it would be excellent practice for you. So let's do the first one and the third one. Okay, and we'll save the second and the fourth one for you. So topic 1.1 is the structure of water and hydrogen bonding. And you've got 2 pictures. You've got a beautiful picture of ice, and it illustrates those water molecules being bonded by hydrogen bonds, and they're actually separated, which is what makes them less dense, and they float in liquid water. And then you've got an image of liquid water. The question is asking you to describe why hydrogen bonds form between water molecules. So go back. Water is made of 2 hydrogens and 1 oxygen. And because the oxygen is negative and the hydrogens are positive, they are going to be attracted to one another, and that is the hydrogen bond. So your response should indicate that the slightly negative oxygen atom in one water molecule is attracted to the slightly positively charged hydrogen atoms in another water molecule, so that those hydrogen bonds form between the 2 water molecules. I'm going to skip this one for now, but you should go back and practice it. It's about carbs. So first of all, you want to discuss the structural components of the group. Then you want to identify 2 examples and briefly describe their function. And then also explain the process that joins these polymers from their monomer subunits. So I'm going to skip that, but the scoring guidelines are here. You can go back to that. Let's take a look at 1.3. This is a question that uses membranes as a vehicle or a way to get you to talk about macromolecules. So you're asked to identify 3 macromolecules that are components of the plasma membrane in a eukaryotic cell and discuss the structure and function of each. And you should be able to do 2 super easily just based on this review. The third one I mentioned, but you may have to rely on prior knowledge for this one. So, and I know this table is not very good, but you can see that definitely we've got our phospholipids, which is a glycerol with 2 fatty acids, and the polar phosphate group is attached. And this is what makes those cell membranes selectively permeable. It restricts some molecules from passing through while allowing others. Cholesterol is another one. It makes the membrane fluid. It's got a 4 carbon ring structure. And then you could mention proteins. And as I just briefly mentioned, they are embedded as integral membrane proteins, or they are peripheral membrane proteins. There's a 4th one for you, but I'm going to let you do that on your own. It's talking about how enzymes are constructed. And the key to this one is to remember that enzymes are examples of proteins. Okay, so quick check for understanding is getting practice with the science skill describe. And it puts it together again with a cell membrane context. So estrogens are small hydrophobic lipid hormones that promote cell division and the development of reproductive structures in mammals. Estrogens passively diffuse through the plasma membrane to bind to their receptor proteins in the cytoplasm of target cells. Describe one characteristic of the plasma membrane that allows estrogens to passively cross the membrane. So you need to understand about the plasma membrane, and you need to understand about estrogens, which are lipids, right? So Because estrogens are nonpolar, they slip right through the spaces between the phospholipid fatty acid tails, which are also nonpolar, so they enter easily. And then just to show you a award-winning full credit student answer, let's look at the top one. Plasma membranes are formed of phospholipid bilayers with a middle region made of hydrophobic tails. Because these are hydrophobic, the estrogens can diffuse between the tails and into the cell. This allows small and uncharged or hydrophobic molecules to diffuse into the cell passively. So that's a full credit answer. So remember that your task verb describe is to give a picture of something in words. Describe it as if someone could not see it. All right, so really quickly with your debrief, hopefully now you're able to explain the properties of water that result from its polarity and hydrogen bonds. You're able to describe the composition of macromolecules and the properties of monomers and the type of bonds that connect them in biological macromolecules and describe biological concepts and or processes. Two key takeaways: living systems are organized into a hierarchy of structural levels that interact, and the highly complex organization of living systems requires constant input of energy and the exchange of macromolecules. I'm going to skip this common errors and misunderstandings slide in the interest of time, but please view it if you stop and view the recorded one. I want to give you your homework. Your homework is due tomorrow, and Ms. Pfizer will be with you tomorrow to go over it. This is a carbon question. It tells you that carbon is very important in living systems, and the following structures are examples of two different categories of macromolecules. You are asked to identify each category, category 1 and category 2, and then describe how each type of macromolecule is important to the structure and function of living things. So I have presented both of these, so hopefully you can easily identify them and then describe their function. All right, so thanks for watching. It was great being with you today. I hope you'll join us again. Make sure you complete that practice question. We'll review the answer in the next lesson. Have a great rest of your day. Bye-bye.